So fructose that was absorbed in the gut from the diet, moving through the small intestine, will eventually make its way into the little veins that kind of line the small intestine, which all drain into this big hepatic portal vein, which takes a lot of the blood that carries nutrients right to the liver. So the liver is the main site for encountering both the new metabolites and the new nutrients that are moving from the gut into the liver, but they're also responsible for metabolizing fructose. Because the liver is one of the only areas with a GLUT2 transporter that allows fructose to move in, and it's one of the areas with the enzyme that's able to phosphorylate fructose. So this is my little diagram of a liver cell. And liver cells on their surface have GLUT2 transporters, which are these proteins that are facilitated diffusion transporters. So they allow fructose to just simply move with concentration gradients from the outside to the inside, typically, depending on whether or not it's a high concentration out here and a lower concentration inside the cell. And if I have a high concentration of fructose, let's say I ate a big fructose containing meal, there's a lot of fructose in the interstitial space, the space outside of my liver cell. Some of that fructose will diffuse through this GLUT2 transporter into the cell. And soon as that fructose is within the liver cell, it's quickly phosphorylated, or a phosphate is stuck on that fructose by the enzyme fructokinase. And kinases are enzymes that facilitate the transfer of a phosphate from one thing to another thing. In this case, we're facilitating the transfer of the third phosphate on ATP onto fructose to form fructose 1 phosphate. In the process, we're pulling off that third phosphate or the gamma phosphate from ATP. Therefore, we're producing ADP. We're turning adenosine triphosphate into adenosine diphosphate. Therefore, the overall levels of ATP within the cell are dropping. So we have an ATP depletion in the cell, and ADP levels are starting to rise. And so this enzyme fructokinase is unique because it lacks feedback inhibition. So typically, when I have an enzyme like this that's responsible for the initial phosphorylation of some molecule that moves inside the cell, like glucose, when I phosphorylate that molecule and I turn it into glucose 6 phosphate, for example, that glucose 6 phosphate will come over and it'll inhibit this reaction from moving any further because I've already formed what I want to form. I've formed the end product, therefore, I don't necessarily want to expend all my ATP to form more of this if I already have some. So fructokinase lacks feedback inhibition. So if I have a lot of fructose molecules within this liver cell, I'm going to phosphorylate all those fructose molecules into fructose 1 phosphate. Therefore, I'm decreasing the amount of ATP. I'm increasing the ADP. So I'm changing the ratio of ATP to ADP, making it a smaller ratio. And then I'm also getting rid of a lot of my inorganic phosphates because I'm taking all my phosphates from ATP and transferring them to this fructose 1 phosphate. So the number or the level of inorganic phosphates is going down within this cell. In the process, because I'm forming all this ADP, it turns on an enzyme called AMP deaminase, which will facilitate this reaction here, where I take two ADP molecules that I just formed because I have a lot of ADP, and I form one ATP and one AMP. So this cell is experiencing an energy deficit. I don't have enough ATP, but I have a lot of ADP. And so the issue is I'm going to trade some of that ADP to form ATP to counteract the overall energy deficit that I'm experiencing right now. But in the process, I'm forming a lot of AMP, adenosine monophosphate. And the formation of AMP will turn on this enzyme right here, AMP deaminase, which breaks down AMP into inositol monophosphate. And that starts what's called the ADP degradation pathway or the purine degradation pathway. And this is the rate limiting step typically. And so ADP is turned into AMP through this reaction right here in order to boost my levels of ATP. So it's a compensatory mechanism because ATP levels are falling within this liver cell. And then that AMP is converted into IMP, eventually inosine, eventually hypoxanthine, xanthine, and the end product is uric acid. But in some animals, in some mammals, I can convert uric acid into a molecule called allantoin. But in humans, we've lost the enzyme that converts uric acid into allantoin, so this is no longer possible, and the end product is uric acid. And so this is the structure of uric acid. It's what's known as a purine because it has a general structure that resembles purine rings. It resembles adenine, which is this structure right here. And uric acid is implicated in a lot of different issues because it's able to crystallize in joints, forming gout. It has a role in causing some blood regulation issues and causes an increase in the overall pressure within the arteries. And some of that has to do with its inhibition of nitric oxide synthase within endothelial cells and also the stimulation of vasopressin. And then it causes, or it's implicated in the causing of non-alcoholic fatty liver disease. And that may be more of a result of fructose. It's implicated in metabolic syndrome, type 2 diabetes, and chronic kidney disease. So typically within the serum or within the blood, the concentration of uric acid is around 3 to 7 milligrams per deciliter or 180 to 420 micromolar. But with animals or with mammals that have the enzyme uricase, which is able to break down uric acid into allantoin, it's much lower. But the issue is the solubility of uric acid really decreases at around 6.8 milligrams per deciliter. So if you get above that 6.8 range, you start to form these precipitates that form uric acid crystals, and most of those crystals will form within joints, and some will form within soft tissue, especially in the kidney. And within those joints, the issue is if I start forming these crystal lattices of uric acid, I have innate immune cells like macrophages. Say this is my macrophage that has the ability to recognize these uric acid crystals within joints, and it detects those crystals, and then it then releases inflammatory cytokines, which recruit neutrophils to the region. So typically, it'll release a signal molecule called interleukin 1 beta, which will signal for neutrophils, another innate immune cell, to come into the area. And neutrophils are typically pro-inflammatory, so they'll really inflame a joint. But the issue is fructose can really increase the concentration of urate, which is the form of uric acid that loses the hydrogen ion in the serum by upwards of 0.5 to 2, depending on the amount of fructose ingested. And so there's this question, why did some primates lose or why did they lack the uricase enzyme? And that has to do with the fact that in those sort of seasonal, more northern environments, there was a really strong drive for these primates to store fat. And uric acid, this guy right here, plays a large role in prepping primates to store fat and enter kind of almost like a hibernation or a form of torpor that allows the primate to survive these prolonged periods of famine that were common to ancestral hominins. So there's a few issues with uric acid. One of them is it's when I'm forming uric acid, this enzyme xanthine oxidase will form superoxide and hydrogen peroxide, which are considered free radicals. Hydrogen peroxide is not a free radical, but it can react with superoxide to form a free radical. And then uric acid can also cause these enzymes called NADPH oxidase enzymes that are commonly within the plasma membranes of hepatocytes or endothelial cells or adipocytes or immune cells to be stimulated or upregulated. And then it can also influence the inflammatory state of a lot of cells when it's moving through the cell through these transporters, which are sodium dependent. So they just move, sodium will enter the cell and uric acid or urate will follow. Um, and overall, it's causing immune issues, and then it's also suppressing AMPK. So it's causing kind of metabolic issues where the cell is unable to really compensate to that ATP depletion because AMPK is involved in a lot of the reactions that facilitate the cell being able to produce ATP. So this AMPK enzyme's turned on during exercise, turned on during stress, but uric acid suppresses it, so ATP levels are kept low within the cell, which is an issue because fructose metabolism already keeps ATP levels low within hepatocytes or liver cells. So this is just a quick diagram that shows how this enzyme xanthine oxidase right here, in the formation of xanthine from hypoxanthine, it's turning oxygen and it's depositing an electron on oxygen to form superoxide and hydrogen peroxide. And superoxide and hydrogen peroxide can react and tend to form hydroxyl radicals, which are extremely reactive, and they tend to be diffusion limited, where they'll just diffuse and the first thing they hit, they'll react with because they're so likely to deposit that electron on something. And then xanthine is converted into uric acid, further producing more of these superoxide and hydrogen peroxide. And then uric acid, as you can see here, in humans and higher primates, um, they've lost the ability to convert uric acid into allantoin. So uric acid can be excreted in the kidney, can be excreted in the GI tract to some extent, but it can also crystallize if the solubility or the concentration exceeds around 6.8 milligrams per deciliter. Then it starts accumulating in joints. Here's another image of that enzyme xanthine oxidase and the formation of superoxide anion and hydrogen peroxide in the process of producing xanthine, which is one of the precursor molecules to uric acid. And then here's a picture of the membrane of an endothelial cell. So endothelial cells are the cells that line blood vessels, endothelial cells. And on the membrane of these cells, there's an enzyme called NADPH oxidase, and there's a couple different isoforms. This is one of the isoforms, so NOx2, NOx1 is another isoform, NOx5 is another, NOx4 is another. But uric acid can actually upregulate the amount of these NADPH oxidase enzymes. And the issue is each one of these produces free radicals. So extracellular uh, ROS is just another term for free radicals that are moving into the space outside of the cell. And the issue is some free radicals is normal, some production is normal, but excessive amounts will lead to kind of a strange way that the cell's signaling to other enzymes within the cell or enzymes outside of the cell, which can cause some pathological conditions, including the disruption of the enzyme nitric oxide synthase, which is really important for producing nitric oxide, which tends to dilate blood vessels when they experience shear stress. And so that's part of the reason why uric acid is implicated in hypertension and other issues. So the overall theme is that because of the metabolism of fructose, fructose will enter liver cells, cause ATP depletion. Because there's no feedback inhibition for this fructokinase enzyme, so I'm turning all my ATP into ADP. And then that depletes the ATP within the liver cell, causing the formation of uric acid due to this purine degradation pathway being turned on because of low ATP and high ADP. And eventually, That leads to the formation of uric acid, which, if kept at normal concentrations, actually serves as an antioxidant. But if it increases past this level right here, it goes past its solubility point and it starts precipitating in joints, and then it can also have effects on cells